I'm very, very pleased to have Amanda Johnson here from Margaret Gardell's lab at University of Chicago to tell us some really exciting stuff about cells and morphogenesis and gastrulation. So why don't you just take it away?
Cool. Thank you. Thank you for, oh, sorry, my voice is very loud. So if I need to move this down. So I'm a fifth year PhD student. So I'm in the Gardell lab. I'm in the development and regeneration stem cell biology program, which is a long name, but DRSB for short. And so I'm interested in human development, mainly how these really migratory cells control early gastrulation. And so I think if we think of a cell and like the mechanics of how a cell actually moves, we know a lot about, the structure of the cell. So the actin cytoskeleton, we know they generate forces. We know that there's polarity proteins that kind of help reorganize the actin cytoskeleton. And we know that there's a bunch of adhesions that can form and they can attach to other cells and to things. But I think one of the questions that really interested me when I started my PhD project was if we know how these cells move, but we don't really know how they know where to go and where they end up. And so that was something that kind of like drove my project. And so I think, we saw a lot about fully formed organisms, but before the organism forms, development has to occur. And so if you're a developmental biologist or you like developmental biology, you like to watch embryos form over and over again. And sometimes if you look down at the cell level, it looks super chaotic, but somehow every time an organism forms and you have all these tissues that are doing their job in that. And so what I was really interested in is this early process of gastrulation. And so a couple weeks after an embryo implants, you have these three tissues that form, which are this middle red tissue, which is mesoderm, ectoderm, and endoderm. And what's cool about them is that these three epithelial layers will generate the entire body form. So from these three tissues, which we call a trilaminar disc, you get all the structures of the body. So anything from your heart, your skin, your bone, your muscle. And so this entire process of reorganization and generating these three tissues is really driven by these mesoderm cells themselves. And so if you look at a top view of a human embryo, which kind of looks like a disc shape, and if you think of these red arrows as the mesoderm cells, this process really gets driven by the dispersal of the mesoderm cells themselves. So along the embryo, they migrate and kind of fill up the space in between the ectoderm and the endoderm. And so that really it looks a little bit chaotic, but in the end, dependent upon where those cells end up and where they began is kind of determine what cells they'll end up as. And so depending on if it's more posterior or more anterior in the embryo, you'll get cells that are going to be the muscle cells, or you're going to get like heart and head muscle cells. And so this organization and dispersal where these cells end up is really critical to how these tissues actually form. And these whole organisms are kind of difficult to disentangle kind of because there's a lot of other tissues around it. We also can't really study human development post-implantation because of, I guess, obvious challenges of not being able to look at an embryo after it's been implanted in a human. So in the last decade, there's all of these kind of organoid models that have come out to try and understand these processes. What I really liked about them is that they're really simplified. And so you can remove any kind of complexity that you are kind of like confounding variables to try to understand what's happening. So you can decide what kind of tissue level or cell level dynamics you want to understand. And then you can kind of pick and choose your poison, like what kind of organoid you'd like. And I was really drawn to these rigid micropattern organoids. And so basically I can control the geometry and shape of how I plate cells, and I can differentiate them into the three germ layers. What I thought was really cool about this one in particular is not only that you can control the shape and the size, you can image them on a normal confocal microscope. It also takes away the extra tissues that you aren't really interested in, and it contains the mesoderm cells, the ectoderm cells, and the endoderm cells all within it. And so you can also give it this one signaling molecule BMP4, which we know kicks off gastrulation in humans, and they will self-organize over the process of 48 hours. 48 hours is also like an awesome time frame for a PhD project, so I don't have to wait like 9 days for something to form. So that was also like a side plus. And then I liked how reproducible it was and how high throughput it was. So you can get, this is like one corner of a glass cover slip, so you can get like tons and tons of these. Depending upon what lab, everyone will, as long as you give it the same concentration of BMP4 or even varied a little bit, you'll still get this same reorganization. So to me, it was kind of like a genuine self-organization process that happened. That was in vitro, but also kind of had some dynamics to it. And so one of the things I based my entire PhD project saying that these mesoderm cells actually migrate in these organoids, and I actually never knew if they did until I finally got a cell line that that reported like the mesoderm gene, so I could live image them over time. So I did a bunch of fixin stains, and I was telling my PI, I was like, okay, yeah, these cells are migrating, but I'm basing this on the fact that I see them moving increasingly inward, but at fixed time points. So I finally got the cell line that's tagged with GFP, so I can watch the mesoderm cells move during the differentiation process. So over 48 hours, they kind of start to turn on the mesoderm gene on the edge. And if I stare at a lot of these movies many times, like I have for like an entire year straight, at first it was like confusing to me because they look kind of like ants, is what I was thinking of. But then it seems like there's this order that kind of erupts where there are these cells that instead of staying in this little, I call it like a little ant annulus niche kind of thing. That's my non-scientific term. If someone has a better one, let me know. There were some of these cells that kind of escape and like move in towards the center, which we know happens in development, which we're basing on like chick embryos, which have a very similar geometry to humans. We also know from fixed samples of human embryos at this stage in development that these cells are filling in the space underneath. So if you look at a side view of these cells, there's these mesoderm cells that are marked in red. You see them when you fix them at the end of the differentiation, you have them kind of looking like they're going underneath those blue cells, which the blue cells are the middle cells, the ectoderm or the epiblast cells. And so I really wanted to look at all of these gastrulase differentiating. I wanted to track their single cell tracks over time and kind of understand their dynamics. And so see if there was any kind of order or regime that could be recapitulated that I could kind of dissect what's going on. Because again, we can't really limit. And even in whole embryos, it's kind of hard to image live dynamics, especially because you need like specialized like light sheet microscopy and then to extract the embryos itself. And so what I did for a long time was, track these individual cells to see what they're doing. So we know they increase their speed. They increase their speed to around the same speed as we know mesoderm cells in mouse embryos migrate, which is on 0.6 microns per minute. So yes, they're motile, which was exciting for me because, I took the risk basing my whole PhD project on something that I didn't know was going to happen. But what was confusing to me is I know they move inwards, but if you try and correlate the actual inwards movement, so they're biased towards the center, it doesn't really correlate with speed. So to me, that meant if they're increasing their speed, if they're moving inwards and they're not moving inwards because they're increasing their speed, there's got to be some kind of other mechanism for them moving inwards. And so that was something that drew me forward. So I wanted to know, why does the speed not explain this direction of these cells? And so if we look at a cartoon schematic of a gastroid, my idea was that you have this motility that increases, and then you have these little ant annulus, they're swirling around each other. So their motility is unchanged, they're staying fast, but then some of these cells kind of move inwards. And so I was trying to understand why does that happen pretty much. And so there was this one paper in zebrafish that knocked out this signaling gene, the signaling receptor ligand combo, and showed that there's kind of like a bladed mesoderm migration during gastrulation in zebrafish. And I thought, okay, well, maybe this is something that humans might employ. And so the first thing I did was I turned to fixin stains because I wanted to see if it actually was expressed. And so in humans, I had no idea if this apelin receptor was expressed at this gastrulation stage. And so it was cool to see that not only it's expressed highly in the mesoderm cells, but it's pretty specific. Thank you. Pretty specific to the mesoderm cells themselves. And so the green is this receptor. Basically, what this receptor is just the ligand for this receptor was only discovered in the last 10 years. So there's not a lot of literature for me to go off on it. But basically, I see that it's expressed in the gastroids. And so as a developmental biologist, I was thinking, okay, what if I perturb the receptor? What's going to happen to the dynamics? And so what I see pretty consistently is if I take the ability of those cells to respond to the signal, you remove that ability of those cells to break free from their little ant niche and move inwards. And so they kind of stay stuck in the center, on the outside annulus. And so at the end of the movies, if I live image them, track them over time, and if I see at the end, the organization looks different, where you have these cells on the outside that are kind of stayed near the edge versus the normal, wild type situation where they're moving inwards. But interestingly, it doesn't really decrease their speed, so they're still migrating fast. And so, fast in terms of cells. And so it doesn't really explain why they're not moving inwards, because there still are kind of moving around, kind of like swarming on the outside, but they're not moving inwards. And so I can try and take those single cell tracks and kind of look at their behavior and try and understand, okay, what's going on that they're actually not moving inwards. And so if I take all of the tracks of the gastroid in the knockdown situation and the control situation and plot the ones that move the most inward to see what kind of characteristics those tracks have. This is like what I have so far. If anyone has better ideas on how to like understand different movement types in vitro, please let me know. But visually, they look kind of like they're lost, right? Like they're kind of just like zigzagging around. They don't ever break free and like move towards the ectoderm cells.
Sorry, I'm really confused by you used the word speed. You mean so you track the centroid of one cell and you ask what's the mean displacement over a given increment in time, and that's what you call speed. But what you call displacement of is like a population quantity. Is that what I should know?
Yeah, speed is just like the instantaneous speed. Yeah, frame to frame.
Per cell.
Yeah, per cell.
Okay. But
yeah.
The thing that you're talking about with like this translation is like something about like the mean position of all the cells.
Yeah. Okay. You mean like well, with these it's just like the entire track. But yeah, for me it's like the speed is still, or the frame to frame speed, the instantaneous velocity is similar if I mean them all over time. But then yeah, their distance traveling inside is less, I guess is what how I think about it. Which again, it's been hard for me to like figure out exactly how to formulate it in specific words or ways, but I think it's easy to see at the end point the phenotype where the cells have like not moved inward, I guess. I'm just looking at time. And so one of the other questions was, if there's this directional migration, especially at gastrulation, if we think about gastrulation as a tissue that's changing size and shape and cell number, you have these mesoderm cells that are leaving the outside, and then you have this neighboring changes. So you have these like boundary shifting. And so what I think an embryo needs as it's developing, especially at a gastrulation stage, is not a point source of a signal, but something that can move with the cells itself. And so if the epiblast is changing shape because those cells are changing into mesoderm cells and moving away, and then the mesoderm population is getting larger, how is that continually being controlled over the course of an entire week, which is what gastrulation takes place in humans? And so I think that these guidance fields, they must adapt to the actual tissue geometry themselves instead of just having some kind of static point source. And so also I'll preface this is a work in progress graph. It's probably like not even mathematically correct, but this is what I have on my slide so far. And so what we were thinking is in the zebrafish paper, which is by Andrea Pauly, Edward Honezer came up with a minimal model to explain how the mesoderm cells might generate a gradient themselves. And so we were thinking, if the zebrafish embryos kind of employ this, is this something that the human embryos would? Because human embryos definitely change their size and shape as gastrulation occurs. And so I've been working with Alex Plum, who's at UCSD, and Isa Mateo-Serra's lab, to try and use our model and see if it actually works on the human cells instead of just mesoderm or zebrafish cells. And so what we can take is all of the single cell tracks and get their diffusivity. We can also take their ligand diffusion to decay. So this ligand that the cells respond to is conserved across vertebrates. So the amino acid sequence is very similar. And so we can kind of assume that the diffusion of that ligand across the cells is going to be kind of conserved and similar. And again, like the ligand's only been discovered in the last 10 years. So there's not a lot of literature on it. There's also no way to live image this gradient because it's a secreted peptide hormone. So the modeling is like a very nice way to try and understand what's going on with the ligand in terms of the cells. There's also one very recent paper in the last like month or two that came out that might have been able to do this in zebrafish, but yeah, that's another tangent that I could follow, but I don't know if I want to be here for 5 more years. But so, I think one of the things that was really cool about the model is that you can fit this beta, so you can take all of the cells, the leading edge cells that are moving inwards, take the top 95% of those and parameterize this one beta, which kind of explains how the gradient biases motility. And then we can look at the wild type situation, which is the control, and the knockdown situation, and see if we can actually predict the density fields with that one beta. And so we see that it can. So we can take this single parameter link that density organization to the actual migration. And so it's a self-generated gradient while we look at this because of the fact if we were thinking of something that had one signal source and the cells were just responding to it over time continually and not depleting it, the density of the cells or the consumption of the cells wouldn't really change how the ligand is. And so basically what we would see is how I understand it is that you would just have a bunch of cells that are at the edge, at the bottom, moving inwards. And so the cells consumption is shaping the actual gradient density of the cells moving themselves, if that makes sense. And that's the best way I could describe it so far. And so one of the things, this is like a very recent work in progress still. So it was cool to me that the numbers worked in the humans and that you can actually recapitulate experiment and the knockdown with the same beta. And so I thought that it was like suggesting that human cells actually do have this self-generated gradient that they're doing in vivo. One of the things that is really cool about the gastroloids to me is that you can change the geometry, and which would alter the signaling gradient. And so in an in vivo embryo, you're kind of restricted to actually what the embryo size and shape is itself. And so I think that one of the cool things about the gastroloid is that I can change how large it is and how small it is, which would in turn change, you know, that ligand concentration source. And then also you can change the amount of cells that are accepting that ligand source. And so if anyone has a cool geometry, that would be cool to look at with like a self-generated gradient type model. You can pretty much do whatever. I have like donuts, donut triangles, I call them donuts, which is like normal donuts. Yeah, you can pretty much make anything with gastroloids. And so they do kind of cool things. But I'm trying not to get too sidetracked, I guess. And then another kind of avenue that I've been looking at, so discussing with, Alex on the self-generated gradient model, I think most of the migration, according to, the paper and him, is due to this, chemical diffusion, self-generated gradient. But I think there's also mechanical constraints provided by the ECM. So those outside cells secrete a high level of fibronectin. And so the mesoderm cells secrete laminin. And so there might be also some mechanical constraints. So some kind of cell sorting mechanism, based upon differential adhesion. So I think that's something that I haven't thought about yet. And I've only been basing this purely on like a signaling self-generated gradient. And then one of the cool things that I've been doing right now is seeing if this migration impacted fate. And so now that I have this phenotype where I've kind of taken away those cells that move inwards, can I take single cell data and see if those fates were actually impacted? And so, but also that brings other challenges like, was that signal actually important for that fate or was it the migration itself? And so that's something that I will have to think about. So instead of, maybe that, maybe Aplin is important for fate acquisition, but migration is not, or something like that. So that's something that I just got the QC approval for my single cell, which took like 3 months to get this experiment to work. So that's super exciting. Hopefully I'll have an update in the next 6 months or so on that. I had no idea how long this was going to be. I'm sorry if it's a little bit short, but I'd like to thank my lab, obviously Alex and Mattia, for being very nice and explaining everything in detail to me. And there's a heart for Valentine's to say, I guess.
Means we can ask you ample questions.
Don't worry.
Yes.
I have one, which is you mentioned like kind of gradients of adhesion as like cell sorting, which I'm like aware of, and so I guess in the sense of like, is part of why like you're thinking this because you see those like cells like peel off from the rest kind of on their own, and they're not, because it looks like they're not really moving as like a united front. It's like you have these like individual guys that like go and peel off from the rest, which means they have to like detach themselves, right? In a nice way.
Yeah. I mean, sometimes, which is very annoying for imaging in my data, they do peel off and like curl over. But so Margaret is very obsessed with this like shrinking in the beginning. So like the cells kind of look like they're all shrinking together, which I guess is the only way to explain it. Which I think could be this is like a wild hypothesis. So there's this paper by Amy Shire who looks at like BMP and response to fibronectin deposition. And so I think since I'm giving this system BMP, it might cause like secretion of fibronectin on the edge cells just in response to BMP. And so maybe that like is contributing to some pushing a little bit of those. There's also another Diana Pinheiro in Austria who's studying these cells actually on the outside get super big. You can't really see it here, I guess, but these yellow cells. And so one of her hypotheses is that those cells are getting very large and they're also pushing those cells. So I think there's some mechanical constraints, but that's why the escaping inward in the apelin signaling is like, I don't think anyone can argue. I mean, hopefully not. I don't know. You can argue with me. I could be wrong.
And just to clarify, so the ones that we're seeing here on the previous slide, the color, I guess, for like the ones that we're seeing here are like the middle ring on the previous slide. Right?
Yeah,
there's cells on the outside and there's cells on the inside.
Yeah. I have other data where I can live image those blue cells. They're both in GFP, which is like very annoying. Ideally, I would mix them together and image them together, but yeah, they're both GFP, which is so I kind of like compare their speed. So the speed of those middle cells is like 0.2 microns per minute, which is just like a similar speed to normal stem cells. So it's nice, and they also don't have a directional bias, which is nice as well. So basically, the mesoderm cells are the only cells that have a directional movement inwards, it seems.
Vicky had a question.
Are you able to image your cells at a high enough resolution to maybe see any like directed protrusions that would further indicate that this is like actually an active migration? And then also, would that maybe change if you like knock out the apelin receptor?
I had this crazy idea. So one of the other things I really like about the stem cell system is you can mix cells. So I did this really low, I have actin tag cell lines. And so I mixed like 1 to 20 with wild type so I could see the actual actin cells moving in. But it was hard to get high enough resolution without putting a lot of laser on them and killing them. But ideally, I have like some fixed samples. But yeah, otherwise it's an entire mess because it's like all on top of each other. I don't know whose actin is who. Who's acting up? Just kidding.
So very cool. Like I would love to ask if it would be fun and profitable to take your use of the word diffusion very seriously. So like if these were like Brownian pollen grains, you would be able to write down this like fluctuation dissipation relation, which is like, oh, the degree to which you are noisily like buffeted by the bath is also related to how quickly you will move with an intentional kick from a force.
Mm-hmm.
So if you take your beta terms effectively per cell and you compare them to their effective diffusivity, do you on average see that like the most noisily jiggling cells are also on average likely to be the ones that have moved farthest?
I think some of the things I think we do have in some other, but I don't know exactly what is there, but I think, I mean, it's very hard to track these cells, especially I feel like the tracks in this beginning annulus are not as accurate, especially because they're all like swirling all over each other. I mean, I spent like a very long time trying to like make sure kind of that they're accurate. And so sometimes I get a little nervous like comparing them, but like the outer annulus versus the inner annulus. But that's something we were trying to figure out, like how do I compare the actual behavior? Like they look like swarming ants, right? Versus are they swarming at the beginning or do they already have some kind of directional? And where do the directional points start? Yeah. That's something, I'm still working on. I could talk about it if.
Can you sparsely label can you only label a few of these cells with GFP and then the rest of them have not GFP and then get better tracks that way?
Yeah, I've done a 1 to 10 ratio and they kept peeling off and I was like, it's frustrating. But I want to do it again though, because maybe I got a little too crazy with the mixing because I was like, I want to see one cell divide and then those 2 cells divide and like track those. But yeah, maybe like the ratio, sometimes they really don't like being mixed. And then so they'll like get all, I was also mixing iPSCs, which is induced pluripotent stem cells and embryonic stem cells, which because I didn't have the right cell line at the time. So now I think I should try mixing embryonic with embryonic because maybe the induced and the embryonic mixing was like, they weren't very happy with that.
I'm gonna ask like kind of a naive question, because theorists' favorite thing to do is to ask a person to do more experiments. So like if you could design an experiment where like if you could like put like a wall and like if you could have like a long sheet or whatever of these cells? And like, if you could, is there a way that you could force one side of it not to differentiate? Or is it always going to be like symmetric like this way?
Oh, so when I was at, that was like what? So when I was at the MBL last summer, and so there was this lab that has this way to take stencils and put them on top of why am I forgetting the word? Not a strainer. I guess it is a strainer, yeah. And so you can put a different signal on top versus bottom, so that could induce whatever you wanted in the stencil. That was like a lot of excitement, and I still haven't done it yet because it's hard to image, but you can. So yeah, that's the pipe dream. When I first started the project was to create an isolated source of these mesoderm cells, so kind of like induce a primitive streak like what you'd see in an embryo. So if I could have like one point source of BMP on one edge, which is create asymmetry. But yeah, TBD. It is possible though. Technically challenging, but there's also experiments that, I mean, I did this in my master's. There, you can put beads soaked in a growth factor. People do with like chick embryos, but I don't know how to get the bead to stick on the cells. But it's easy to stick on a chick embryo, yeah.
Cool. Thank you.
You mentioned the geometry problem at the end. So epithelial cells, at least in the eye, they form like a tight monolayer of hexagons. If you actually dissociate the cells, they turn into these snowman-like things with like a belt in the middle. So you have hexagon on one side and the basal on one side, and they kind of regrow out and somehow sense that the monolayer is complete and stop growing. I don't know the math behind any of that, but I think that would be a cool feature to study in something like this.
Yeah, like
do for the next 5 years.
yeah, like dissociate them and see how they
like grow out again, because it goes from like a complete snowman type thing into like a hexagonal monolayer again.
There is one paper that dissociates the gastroids once they've been differentiated, and they all they sort themselves into their little like the mesoderm cells, and then but they didn't look at like their contact or anything, so like how they actually formed, just that they formed with each other, and then the cadherent expression. But yeah.
Would it be like incredibly bad to like laser away like half of this after it started?
Oh yeah, like or scratch it or something.
Yeah,
something sharp. I think they did that for like ERK signaling, because it's like a wound healing response. But yeah, so one of the things that I'm trying to figure out is I can actually take all of these cells and make all of them mesoderm cells. And differentiate them so I can kind of isolate their dynamics of just the mesoderm cells themselves. That's what I've been working on with Billy. And so one of the experiments that I've been planning, I haven't done, is a wound healing. So scratching it and seeing if their polarity is different in the knockdown versus not the knockdown, because we want to see it fill empty space and see if they have like, I think that was, maybe that wasn't answer your question.
No,
but
thank you. Excited to see it. Okay.
Let's thank Amanda.
Yeah.